Now, Forward, the campaign for UBC, is the biggest fundraising and alumni engagement campaign in UBC's history. And with the support of alumni, donors, and friends like all of us joining today, UBC is advancing healthy lives, a healthy planet, and healthy societies while addressing the immediate affordability needs of students at UBC. And today's topic is representative of one of the many ways that UBC is undertaking research to help create healthier lives. Now, today's event, the Innovative Thinkers webinar series, is part of the faculty's alumni programming created to highlight our researchers and faculty and alumni from the UBC Faculty of Pharmaceutical Sciences. The faculty's vision is to be a catalyst for change in pharmacy practice and in the pharmaceutical sciences. Our goal as innovators in pharmacy education, research, and practice is to really support the optimization of drug therapy in the pursuit of improved patient outcomes. And we're all very thrilled to have you all here today with us for this first webinar in the Innovative Thinkers webinar series. Now, I'd like to introduce our fantastic moderator for today's conversation. Let's go ahead and have Dr. Shukli join me on screen as I introduce her. So Dr. Kathy Shukli is a clinical assistant professor at the UBC Faculty of Land and Food Systems and is also a clinical veterinarian at the UBC Animal Care Services. And her research and clinical practice interests include research ethics, the relationship between humans and animals, and practical ways to improve the emotional experience of animals involved in animal research. So welcome, Dr. Shukli.
Thank you very much, Dr. Ross. I'm delighted to be here to moderate this important conversation today. So in just a few minutes, I'll introduce Professor Collier, who will begin with a short presentation. Following that, I will be starting off the conversation by asking Professor Collier a few questions, and then I'll address questions from all of you in the audience. To facilitate this, we will be using Slido, our online audience engagement platform. And to enter your own question, please go to slido.com, slido.com, and use our event code, which is replace, to sign in. And there you can submit your own questions, and you can also vote up other questions that you would like to see answered. So for today, drug testing on animals has been a long and contentious subject. To address animal welfare and improve outcomes, UBC Professor Abby Collier is leading scientific research in modeling and simulation to reduce, replace, and refine animal use in experimental research. Professor Collier is combining lab work with computer modeling to minimize animal involvement in experimentation while improving drug and chemical safety and effectiveness for humans. I'm thrilled to have her here with us today and to have the chance to dig into her impactful research. So let's go ahead and have Professor Collier join me on the screen as I introduce her. Hi. Professor Abby Collier is a professor of drug metabolism and pharmacokinetics here at UBC, where she teaches pharmacology to undergraduate, graduate pharmacy, and medical students, and maintains an active research lab. She's also the winner of the 2021 Center for Alternatives to Animal Testing Award for her scientific research in modeling and simulation. Welcome, Professor Collier. We'll turn it over to you for some opening remarks.
Hi, everyone. Thank you, Kathy and Colin, for having me, and thank you to UBC for providing me with this really important opportunity. So if I might share my screen. Apparently host disabled participant screen sharing. Okay, so I have some slides that I'd like to show you. Perhaps somebody can pop the slides up. Here we go. So this is the reason I've been asked to talk today. And we do a lot in my lab, but one of the really big issues and things that we've been working for since probably 2006 is to reduce, replace, and refine animal use in experimental research. And I'd like to maybe take the audience through some of what is traditionally thought and then contrast that with how we do things. So you're probably used to thinking about a treatment and a control group, and then there's a bunch of mice, and they may not be mice, rats, animals, and then you do something, you give them and you watch them. And in the case that I'm going to show you on the slide, the treatment control group, 3 of them out of 10 survive. That's more than an LD50. And then all of them in the control group survive. It's a very blunt instrument. It's really, I mean, it does as well as it could have done in the time that it was given and worked out. However, what happens here is then we take the same drug, we give it to people, and we put it in clinical trials, and we say, well, you know, the animals have taught us something. I personally believe that if you don't believe in animal research, that's okay. I also personally believe that if you do believe in animal research, that's okay as well. Because in my lab, we've learned this. Instead of taking, and I will go back, instead of taking 20 mice, 10 in each group, for an outcome of 3 and 10, what if we could take, for one drug, what if we could take 5 mice, and instead of putting them through anything, we don't give them a drug, we don't expose them to a chemical, we don't do anything bad to them, but we do sacrifice the mice. I'm not going to lie to you people, the mice don't make it, but when we sacrifice them, we take their major organs. Now, for me, the major organs of drug metabolism, the ones that work out what gets around and what comes up and goes in, are the liver, the kidney, and the intestine. So what if instead of 20 mice for one drug or one chemical, what if we could take 5, harvest their livers, kidneys, and intestines, somehow preserve them, and instead of that one drug, we could take bits and pieces of them and put all of these drugs against them. We do it biochemically on a bench. And then here's the hard bit. We use a bunch of math. That's what we do. We take what we know about the mice and we compare them to humans mathematically. And we know everything about the enzymes. We know how the enzymes work. We know their rates and their controls. So why can't we move this up into humans? And that's what we use to guide all of these drugs in human trials. So there's a few issues here. How well does any single animal model a human? And what I used back here were mice. And that's the most common. Mice and rats are by and far away the most common in academia and also in the drug companies. But there's also rabbits, mini pigs, dogs. So we have to ask ourselves, how well do these animals even model a human? Should we be using them? I'd like to ask you whether or not my answer, instead of 20 mice for one chemical, I can use 5 mice for a dozen chemicals. Exponentially less. However, do I still know whether or not it's good enough? Do those mice or those rats or those mini pigs or those sheep or those horses, those dogs have the same enzymes and transporters? The other thing we need to ask ourselves is, if we're going to use an animal as a model, how much does the liver, the kidney, how much blood flow, does it look the same? And also, how does it vary? Is age the same or different? Sex. We call it strain in mice. So, for example, if you're a horse person, do you have a quarter horse or do you own a warm blood? If you're a sheep person, do you have a Romney or do you have a British white sheep? Like, what sort of pig? These kind of things may make a difference. And so if we're going to use that kind of an animal and translate it to a human, how relevant is it? And then with what I'm proposing, with all of this, yes, of course, 5 animals, not 20. Dozens of drugs, not 1. But how accurate are we? I mean, I'm proposing to use maths to make things work. Do we know what these scalars are? I mean, basically, if we take a little piece of a liver from a mouse that's given its life for our research, how good are we at working out how much we can move that upwards into humans? And do they vary by age, sex, ethnicity, and genetics? And how complex of that can we make it? Because one of the really important things that I have learned in the last probably 10 years is there's a lot of people that know a lot about math, and there's a lot of people that know a lot about biology, but these people don't always talk to each other. So too much biology and the math people don't get it. Too much maths and the biological people don't get it. And we have to talk to each other. And in medicine and in life, we have to make things available. The point being, some of you that don't agree with animal research in any given way, shape, or form might ask, wouldn't you do that in humans? And I would say, yes, we would. And I've been doing this since 2007. So what if you could get people, and much like the mice or rats, you could take out their livers, their kidneys, their intestines, and what if you could put them in a test tube? And what if you could test each and every one of these chemicals with humans and do some maths, and then you can head things into clinical trials? Yes, we can. And yes, we do. Most of the time, this is limited to very specific things where we know a lot about them, or very specific sectors of the human population, such as children or adults, like special populations, where we know we have to be very careful. So we'll put the extra effort in. One of the things I'd like you to think about is, that's a great idea. Let's completely remove the animals. That's replacement. Completely replace. We don't have a mathematical equation for the human body yet. No, we don't. But what we do have is a number of ways of making it work. The big question here is, if you don't want to have animals used at all anymore, how and where do we get human tissues? I'm quite willing to share with this audience that I set up an organ donation bank when I lived in Hawaii. And the reason for that is Hawaii has a hugely ethnically and genetically diverse population. And so a lot of the time, there's not good organ donors. And if there's not good organ donors, the only organs that can be sent from Hawaii to mainland USA are kidneys. Kidneys can last in what we call hypoxic ischemic time. They can last long enough to send them to Oregon or Washington or California. 5.5 hours on a plane, assuming you get it out fast enough. The rest of the time, some of these people died and their families wanted them to be organ donors, and they had normal tissues and there wasn't something for them. And I turned up and I said, can't we use this instead of animals? Can't we take these human tissues and preserve them? And then there's a number of issues there. How well do these isolated human tissues model a living human? What if your kidney and your heart and your lung and your brain aren't working at the same time? Is it even real? So those are questions we try and answer. And we have the exact same problems that we have with the animals. How accurate are the math scalars? If I want to take a piece of tissue from a human liver and do some research and study the chemical or the drug, how on earth do I make sure that that's real? How do I know that the sex or the age or the ethnicity or genetics of the person doesn't make a difference? Well, I don't, unless I do enough experiments. I'm here today to talk to you about how we do things better. I strongly believe that we don't need to do this anymore. In some cases, it's necessary, but those cases should be made. The case should be made because we can do this. One of the questions that I'm going to be asked, and I'm absolutely sure of it, is why doesn't everyone do it? Well, can you imagine having the skills to work with the mouse, to know exactly how to pull out, or the mouse or the mini pig or the cat or the dog, to pull out the exact organs, to use the right drugs, and then do the math and model it up and work it out? I'm not saying it's easy. I'm just saying we need to work harder. And the same thing with humans. How do you even make those conversations with a human, with an organ donor bank? How do you even start to talk to people about their loved one can't be a donor anymore, but maybe they can give you the liver of the loved one that died, and you can go ahead and make drugs for the rest of the world. And those drugs rely on a whole lot of math, but we still have to put them into the human population. We still have to do the clinical trials. So those are my thoughts, and those are the things that I thought might be helpful to interest the people listening. And I'd like to send it back to Kathy Shupley to moderate the conversation, to ask people what they want to know and what I haven't explained that Kathy might be able to moderate and make me explain better.
Thank you, Abby. Thanks for starting us off on that very thought-provoking presentation. That was very interesting. So before we dig in here, just a quick reminder to the audience again about how you submit your questions. You can add them in slido.com, and the insert code is replace. Okay, so just to start off, Abby, maybe this is just more, before we get into the details, a little broader question, a little bit about what inspires you and motivates you to do this type of work.
Okay, so I'm just going to be super honest here. When I was 14, I went to my local GP, and he's like, oh, you know, you're pretty smart and doing well in high school. What do you want to do with the rest of your life? And I'm like, I either want to do drug research or genetics. And he said to me, well, you know, genetics is, you've got to be really careful. There's a whole lot of ethical stuff. And he's like, well, drug research, nothing ever happens there, and we don't do it in New Zealand. And I was like, that doesn't mean it's not possible. And I've always just been fascinated and interested with the way that drugs work in the body. And one thing that I think you all have seen Dr. Ross, who's one of our world's best pharmacogenomicists, that we learn that these chemicals don't exist in isolation, and the way that our genetics work with this works as well. So I get to do both, but primarily I'm a drug researcher, and I'm fascinated by the way it works on an intellectual level, but on a personal level, you know, academics are the ones that want to save the world. And I work for drug companies, and they pay me, and there's all sorts of institutional conflicts of interest and things there. But when I come home to my research lab at UBC PharmSci, and when me and my grad students are working hard, and when I'm teaching my pharmacy students and my undergrad students and my medical students, that's what we're doing. We're making it better, and that's why.
Thank you very much. Very, very eloquent. Great. Okay, so the topic today is around the 3 R's, and just so we're all kind of starting on with the same understanding, the 3 R's, replace, reduce, and refine, were proposed in the 1950s, and they've become widely accepted principles for the governance of humane animal experimentation. Can you elaborate for us what each of the R's means? The R's mean. And in your opinion, is there any one R that we should be focusing on more than the others? And maybe you've already highlighted some of that.
Yeah, I think for the audience that Dr. Shupley could probably do it better than me, but I'll have a go. Refinement means if you're going to use an animal in research, you're going to make it better for the animal, right? Just at its simplest form, you're going to make the animal more comfortable. You're not going to do such harsh things. You're going to, if you have to do an experiment, the experiment will be the least harsh and the best for the animal you can. But it's more than that. Refinement is also making sure that animal doesn't live in a steel cage. That animal has interests and it lives. And, you know, and I would like for the audience to understand that we have gone far beyond the old days. You've all seen probably YouTube videos from the 1980s where there were primates and monkeys and dogs in cages, and it doesn't look like that anymore. But we cannot resile from that. We did that. We have to admit that we did that. I didn't, but I am a product of the people that did that. So refinement means making that better, not doing it, not being so harsh. Reduction is a lot of what I do. Instead of 20 mice for one compound, to like, how about 5 mice for a dozen compounds? Or how about exponentially removing how many people you need? And it's not just me. There's a huge number of people since probably the 1990s that have worked out statistical ways. So there's a really, really old school way called an LD50, and a lot of people in the audience have probably heard this. And it means the lethal dose at which you kill 50% of the animals. Can you imagine that? You're just going to keep giving it to them until they die. Crikey, that's not very nice. No, it's not. And none of us want to do that anymore. And part of reduction has involved working out statistical and mathematical ways of doing that, so we don't have to do it. We can just kind of predict. I'm terribly sorry to tell you all that some animals still die in reduction. And then replacement. That last slide, the second to last slide I showed you, where we don't need animals anymore, we'll just take the animals out of the equation. Take the humans, but I am at pains to tell everybody here how hard it is, not even as a person that just is interested in drug research, but like, can you imagine walking up to someone whose loved one has just died and saying, I'm terribly sorry they can't transplant your liver. May I please have it for some potential drug research in the future that might make it better? And I have done this, and I will continue to do this, and that is replacement. But we can't solve the human body yet. We don't know the math. We don't know how to do all the equations and put it together. But we're working on it, and one day, maybe we will. I don't know. I'm a bit scared of the whole computerized AI. Like, I don't think everything can be math and computers and AI. I don't think we can take people out of the equation. And if we do, there won't be ethics and morals, and there won't be love. But I think, yeah, reduce, replace, refine.
Thank you. As you highlighted, your own research is really at the forefront of replacement and reduction. Are there certain types of research that lend themselves to the application of the 3 Rs, or are there limits to when or these can be applied?
Yes, ma'am. I'm quite lucky with that because my scientific grandmothers and grandfathers have come before me, and we have learned that if you take, for example, a liver out of a mouse or a rat or a human, and you preserve it and you make it very, very cold, right? Like we save these things at -80 degrees Celsius. And if you do that, the enzymes within that organ, and I would like to tell you all that when you put a drug or a chemical into your body, you swallow it and it goes into your tummy, and then from your stomach it goes to your intestine, and from your intestine it goes to your liver. And that is the whole job of the human liver. It is a detoxification organ. It makes things good, it makes things bad. With drug people like me, we're lucky because we get to study the liver, and if you think back to the slides I showed, I definitely showed liver, kidney, intestine, right? So for us in the drug industry, we're kind of a few steps ahead because we know these organs are important and we understand how to preserve them. But I feel so terrible for the neurological people, the Alzheimer's people, right? Like, you can't preserve a brain that way, and we just we don't understand how to do that right now. So yes, Kathy, this is exactly the question, that where someone like me can move these things forward, and I can hope and I can work, but I look at my colleagues in the neurological sciences or skin would be another one. We don't understand how. So our dermatologist people, and I'd like to help. I'd like to make it better. And one day when they work it out, they'll come see me and I'll tell them, yeah, this is how you immediately collect a piece of tissue. This is how you immediately preserve it once they work out what they need to preserve and how.
Thank you. I guess related to that, and I see one of the questions in Slido that it also asked this question, the using animals in animal testing or experimentation is kind of it's been a model around for a long time. What is needed, or what are the obstacles, or what is needed to maybe overcome some of the barriers, or what do we need to do to kind of get to where we want to go? How do we move, for example, replacement forward?
I'm going to say something slightly controversial. and I don't mind this, my colleagues and myself have to be told no. If you want to use 24 mice for something, you just got to be told no sometimes, and you got to find a better way of doing it. And better way is usually more difficult and more expensive. And as academics, we can't often do that. Like, academics want to save the world. And I'd like to let the audience know that I work a lot with industry. And if that's a conflict of interest statement, you need to understand I contract and work a lot for industry, including Genentech, Roche, Eli Lilly, these companies. So they can afford to put money into working out how we don't do this. Whereas the academic researchers, all they know, and they came up, they were a PhD student, a postdoc, and they're not horrible people, but that's how they do their research. And I think institutionally telling people no, you can't use 24 mice, probably not even possible. And it's a very controversial statement. And yes, and I would like everyone listening to me and hearing me to understand that you can't just cut the mice, the rats. You can't just cut the animals out. You can't do that. But there's probably a balance there that we haven't hit yet.
You mentioned some of the pharmaceutical industry. What role does the regulatory bodies like the FDA or Health Canada play? Are they thinking along the same lines, or what are your
So here's an
experiences?
interesting thing. So last year, 2022, the President of the United States signed an executive order that said the FDA, that's the Food and Drug Administration in America, does not need to require animal studies to approve a drug. That was, I think, August or September last year. It's about a year old. They are not required to approve a drug. However, they are on the hook to approve the drug. So that means, what other evidence do they have, and how good is the evidence? And some of what I do, some of this using the human kidney and liver, like, maybe that's good evidence, maybe it's good enough evidence, but you've got to ask yourself, if the President of the United States is going to sign an executive order that says you guys don't need to use animal studies anymore, and for those in the audience that haven't heard, previous to this, before any single drug could be put into a human, it had to be at least given to a rodent and a non-rodent model. Now, the rodent model, usually there was a bunch of them, like, somewhere between tens and dozens. The non-rodent model, at a minimum of 5, of a dog or a monkey, or, even then they would still accept it. But that is what was required to, and, you guys have got to understand that we want to make these drugs safe. Can you imagine if we let a drug out into the population and it starts hurting and injuring people? That's my worst nightmare as a person that develops drugs. But we have to balance it out with how much harm do we do to these animals, and how much harm do we do, and what, yeah. So, yeah. Kathy, that is no answer, but that's your answer.
Thank you. Another slide of question was somebody curious about what, getting into this sector, which also relates to just the role of training scientists, upcoming scientists, and what's how has that evolved, and how should we address that? Or are there things that this audience member who may be an upcoming scientist
how do we train our kids? Forgive me for calling them our kids. Like, I don't have children of my own, but every single one of my graduate students I call my kids, and they allow me to do that, and they have their own mums and dads and stuff. But yeah, absolutely. It's moved on a heck of a lot. So I trained in the late 1990s when I did my PhD and stuff, and things were harsh. And I trained in, by the way, I'm a Kiwi. I'm not an Australian, in case anyone was wondering. We like to get that right. But yeah, I trained in New Zealand, and we run off what we call the Oxbridge model, or the Westminster model, very old school, British kind of way of teaching and learning. And even back there and back then, and I spent a dozen years in the United States. I lived in Nevada, and then I worked in Hawaii at the medical school before I came here to BC. So I've been around a bit, and I've learned a lot, and everywhere I go I learn something. And to answer the person's question. We've learned to be better mentors, and we've learned that we don't just tell you and you do, right? Like, that's terrible. How about we work together and we do something together? And particularly in this kind of environment where I'm talking about things that are quite ethically and morally difficult, like, we have a whole lot of things we need to talk about and do with these animals and with these drugs and with these humans. I mean, it's not just the animals and the drugs, right? Like, think about the human, think about the rare diseases, think about the people that are pinning their hopes on this new drug and how much we take on from that. We just want to make it better. And one of the things that I try and do with to bring it back to our trainees, we don't want to put that onto a trainee. We don't want our graduate students or our postdocs to think it's all about you have to make it better, but we do want them to understand that you are making it better. You are part of the solution.
Great, thank you. Another audience question that is it related to the 3 Rs globally, and how, what's your understanding of how countries navigate besides Canada?
It's very different. And as I told you, I grew up in a small town in New Zealand. I did my training in Auckland at Auckland University in New Zealand, and then postdoced at the University of Nevada for 4 years, 9 years at the University of Hawaii Medical School, then here in BC. I have a whole lot of collaborators in the UK. I actually have UK citizenship. I'm English as well. I understand how it works everywhere, and this includes India and China and Brazil and South America. And yes, there are different ways in thinking and ways of being depending on where you come from. And one of the things I think we find really tough is how do we, I am just going to use the Western world as kind of a big global conglomerate. How do we use that concept? And then how do we talk to our colleagues in India or China where they may have a different idea about how they do science? And yes, of course, everybody has ethics statements, and IACUC is the Animal Use and Care Committee, which you have to get approved before you can do your job. And the same thing with human, IRB, institutional, like, but how those standards are applied in different parts of the world are different. And I think we need to acknowledge they're different, but I don't think that it's okay to do, I'm going to use an informal word here, crappy. I don't think it's okay to do crappy things to animals just because we recognize there's different standards. No. But I do think we need to understand that things are different between all of these different, and in the way that the FDA, in the way that the drug agencies, they call them jurisdictions, right? So you can have a drug approved in a certain jurisdiction. Like, for example, Japan is its own jurisdiction. The US is a jurisdiction, as is Canada with Public Health Agency Canada, where we live here, where I'm very proud to be an immigrant too. So we talk about, and the EU is a jurisdiction, and actually the UK is its own jurisdiction now, plus or minus Brexit and stuff. But I think where this plays in, and this idea of jurisdictions, is it's not about how wealthy or how not wealthy a country is. It's not about what your morals are. It's about how globally we see things, and then how we integrate the ideas about how we use animals, how we respect animals, and how we talk about what we do.
So is there a role for harmonization across these jurisdictions?
Yeah, I would love to see that, but my husband is the best person to ask about this. His name is Michael Coftree. He's a very well-known drug metabolism scientist, but he says scientists would rather use each other's toothbrushes than use their nomenclature. So for those of us that do genomics and drug metabolism, and it's the same thing. I would rather let you use my toothbrush, and I would rather than, agree on, and that is the excessive example of how people don't get along. But it's the thing we have to deal with when we talk about harmonization. How do we get it together across all of these borders, across all of these cultural differences, across all of these moral and ethical differences? Yeah, but harmonization would be wonderful. Yeah. I'm just old enough to be a bit cynical.
Challenging. Okay, so here's a question related to how we do science. How do researchers and scientists currently share results from testing with one another to minimize the need for similar animal testing and to be repeated?
So thank you, whoever asked that question, Madam Sir, thank you. I literally sat down with one of my master's students yesterday morning. We were writing a review together and we were going over, here's 28 tables in the review, and he printed everything out. And then we were going over the tables and ticking off to make sure that every single number was correct. Now, why am I telling you this? Because we used to have to do that, right? Like print everything out. How do we, and that was the way we shared things, was you published it, somebody saw it. These days we have archives, and like, so I would send people to bioarchive, I would talk about medRxiv, I would talk about physarchive, and so people, and some of the journals ask us and or make us do this as well. Like if you're gonna publish something, would you please put your raw data up into a database? And if any in my opinion, if any investigator says no, they shouldn't be published and they shouldn't be allowed. And the one time I've ever been so proud of that is just before I moved here. It was 2012, and a student and a postdoc of mine tried to publish something, and one of the reviewers said, "It looks too good, we just don't believe you." And I was not offended by that because I thought it looked too good as well. So I said to them, here you go, here's the Excel spreadsheets, here is the raw data. I wasn't offended. I felt like I need to share. And this, maybe I'm a new generation, or I'm like half and half of the old generation. We need to share, and these archives and putting these data up so other people can look at them, it's really important. And once you know, because so much of what doesn't work isn't published. So once you know that somebody used, I'm terribly sorry for anyone that this upsets, but once you know somebody used 64 mice to do blah, blah, blah cancer study and it didn't work, nobody ever has to do that again if we share the data, if we put it up in the archives, and we're starting to do it.
Great, thank you. Here's a question around different, where is it? Sorry, no, I've lost it. Oh yeah. Thank you for the interesting discussion. How are these concepts modified for various disease indications, particularly systemic diseases?
Yeah, I think that's a really difficult question, and I think there's no simple answer. Hi, that's a politician in me. Most of what we try and do is model the standard state, and that will give us an idea of what's going on in the population. Some of what I do, and you're asking the right person, because some of what I do is what's called special populations. And so for us, special populations in my lab, traditionally, we've looked at pregnant women and children and the elderly. And so we've tried to, but other people that I know that I work with, they look at your special, like if you have a systemic, like rheumatoid arthritis or some of these systemic things, and then they're not what you would consider populationally normal, but they're not abnormal, they are a state. And one of the problems that we have is first of all, how do we get enough people? If we want to study this in people and it's rare, how do we get enough people? And for every person that says yes to a study, there's 7 people that say no. Secondly, if I wanted to put that kind of odd state back into an animal model, I can probably do it. I can probably knock out a gene. I can probably knock out, and Dr. Ross that was here earlier, he specializes in that. He's super amazing at that. So I work with Colin, we would knock out the gene, but is it real? If I go, and also the second question is, should I be doing that to an animal? Should I be knocking that out in the animal? And so it's a really, really good question. It's very complex. Personally, I'd like to see us do it in humans. This is very controversial. Even Dr. Ross is going to yell at me right now. No, don't knock it out in a mouse. No, don't mess around with a rat or a mini pig or a dog. Like, just go to the human population and work it out. But that is so much easier said than done. And instead of costing $250,000, what if that cost you $250 million? And that's what we deal with, right? Like, we want to make it better, but we sometimes can't afford it, even as researchers.
Right. Yeah. Challenging.
Yeah.
Sort of following on that one, just curious, are there, with AI being in the news and so on, are there new technologies on the horizon that you think will have an impact on this work? No.
Yeah, I've thought about this. So my biggest thought about impact on this work is I teach a graduate class in our faculty that all graduate students have to take, right? And it's just a broad-based class, like, how do you learn about clinical trials? How do you learn about animal research? But right at the start, I ask if anyone's doing animal research. And some people put their hands up and some people don't. And then I ask them if they're doing cell culture. And most of them put their hands up. And I'm like, you guys realize that cell culture is animal research? And a bunch of them are like, no, I'm using human cells. And I'm like, yes, you are. What do you put in your media? So for the people in the audience, cell culture involves a number of very unusual types of cells. And what we do is we isolate them, we put them on a dish, and we grow them up, and then we do experiments on it. And kind of human, kind of weird, kind of not. We definitely put them in a very special atmosphere. We look after them. But we can also put drugs on them. We can look at how they work. Like, so imagine if it's a lung cell and you put an asthma drug on it. How does that work on the lung cell? Or a skin cell and you put, a steroid on it. How does it work on the skin cell? So these are things we do. People think, oh, well, it's a human cell. In order to make these cells grow, we use something called fetal calf serum or bovine fetal serum. It is exactly how it sounds. In order to make, and some of you have heard about Henrietta Lacks, you've heard about HeLa cells, you've heard about these cells that grow forever. In order to keep them growing forever, we kill little calves and we take their serum, and that's what we do. And to every single one of my graduate students, I'm like, are you doing animal research? They're like, no, I'm doing human. And I'm like, do you do cell culture? Where's your FBS coming from? That was a long story to tell you the short answer, which is, Kathy, we now are at a point where we don't need FCS anymore. There are cell culture media that don't have calf serum. Despite that, I'm here to talk to people about reducing and refining animals and doing some math and modeling. Honestly, the biggest thing for me is all those, not just the hundreds of labs worldwide that do what I do, it's the hundreds of thousands of labs worldwide that culture cells that won't need to use calf serum anymore. Why aren't we all doing it right now? Well, it's a secret. There's a bunch of very expensive. So basically, when I run my lab, I buy the things that run cell culture in 500 ml bottles, like half-liter bottles, right? And they're $12 each. And then I add fetal calf serum, I add antibiotics, and we culture ourselves. You can do that without the fetal calf serum, and you buy 100 ml bottles, right? So that's half a can, less than half a can of soda. And it has no fetal calf serum, and it costs $400. Because we don't know what's in it, but I guarantee everyone listening, when we work it out, we will publish it and we will share it, and nobody will be killing baby calves anymore.
Wow, interesting.
Here's your answer. You weren't expecting that, were you?
Thank you. So we're getting close towards the end here. So there's quite a few people in the audience who are interested in what role they can play. So how can alumni support your efforts? Also, how can the general public or consumers help support replacement animal testing? What role can they
Well,
play?
I think I'm very grateful for those questions. I think it's really nice that people want to get involved. I sometimes feel like we're just out here doing this stuff on the edge of everything and nobody really cares. And then I get an opportunity like this, and to hear that people really care and really are concerned and really want to be involved is phenomenal. So how do you do? Yeah. So I would say support things. You don't have to support someone like I'm old. Support our students. Like the next generation are the ones that need to learn. If you're in a position to tell your kids, like, do it this way, like that's fabulous. Like, and that they need to understand that, yes, the old ways aren't terrible, but we have new ways of doing things. And to promote this concept and this learning idea, it doesn't cost $1, doesn't cost a cent to promote these ideas. As consumers, don't be reactionary. It's too easy to boycott this and something that, but as consumers, yeah, I do think there's a role to be played for people to say, how are you doing this? How are we looking at this? How do we move forward together? And one of the things that I'm really militant about is I think that academics, scientists, all of us, we've been doing it too far by ourselves for too long, and we haven't partnered with our people, our communities, and I'm from a real small town in New Zealand, an Indigenous town, and it's called Tokoroa. I learned growing up the value of community, and I feel like scientists haven't always grabbed that. And yeah, so how moving forward, yeah, teach your kids, and not just teach, but like make it available for everybody. Like just be open to those conversations.
Great, thank you very much, Abby. So we have about a little bit of time left, so we wanted to give you, and there's a couple of questions still on their chat, but we wanted to give you a chance to provide some final thoughts about what do you want the audience to think about first and foremost as this program closes?
I just want to thank everyone for being here. I thought I'd be talking to an audience of one, me, Kathy, and possibly Colin. And so I think it's really important for us as scientists, professors, whatever, to understand that we have a community, even if we feel isolated sometimes. But by the same token, if our community reaches out to us in good ways, please don't throw eggs at my office window. Please don't come in yelling and screaming at me. But if you've got a problem, come and talk to me, because I've spent half my career trying to work this stuff out, and it's not black and white. And sometimes I'll disagree with you, but yeah, we don't need to make animals martyrs. But sometimes we need animals. That's yeah.
Thank you very much. Thank you, Abby, for giving us the time today to share your thought-provoking work, and it's been really fascinating. And you've been highlighted the challenges and the realities, and that's really important. Your work highlights the importance of thinking about the 3 R's and on all aspects, but also just pushing ourselves and scientific community to be innovative, so that we can continue to expand the application of the 3 R's, and in particular replacement. So thank you very much. That was really interesting. So now we're going to welcome you to turn off your camera, and we're going to welcome Colin back to make some closing remarks.
Great. So I just wanted to provide first a big thank you to Professor Collier, Abby, and Dr. Shupley for such a very thoughtful conversation today. So Abby, thank you so much for all of your outstanding work that I see clearly is improving the way that drugs are evaluated around the world, saving the number of baby calves that we need to use in the future for serum, which, yeah, I agree, it's ridiculous. You mentioned that I might yell at you. I would never yell at you, and I'm hoping that you come to coffee at 2 with some of us. But you also know that for many of the diseases that I work on, really for many of them there is no other treatment available. So we're trying to really, we're trying to develop new therapies where there is no treatment. And for these patients and their families, they are often so keen to volunteer. They want to, I mean, I'm sure they would be glad to take these drugs without any safety testing. But what I really mean to say is that, you know, there's so many opportunities for those types of patients to help them to help move these new drugs forward really early in the drug development process. And I also wanted to thank you for including the insights from Professor Coughtry, who I've learned over the years is someone who is very good at cutting through the noise and seeing things very clearly. And if he sees the challenges ahead, I know that those are real challenges. And I also want to thank all of the audience for joining in the conversation today and for your really great questions. So I'd also just like to sort of conclude by saying I'd like to invite everyone here to join us at the next Innovative Thinkers webinar in this series, and this is titled Unlocking the Power of Open Science for Health Excellence. And this is taking place on October 18th at the same time as this program is being held today. And there should be a link that will come up in the chat where you can get more information on that. And then just a reminder, this webinar series is created to support Forward, the campaign for UBC, which is benefiting research, teaching, and learning across both campuses. And we are the catalysts that can make that happen. And together we will shape a better world. And so with that, on behalf of the UBC Faculty of Pharmaceutical Sciences, this brings our program to a close. So thank you again for joining us for this first webinar in the Innovative Thinkers webinar series. And we wish everyone well and have a great rest of your day. Thanks.